In the mid-20th century, humanity discovered a chemical that could reliably produce this specific biological end state: mass systemic insect death. The agent responsible for this absolute, sweeping efficacy was dichlorodiphenyl trichloroethane, commonly known as DDT. Because of how it was applied, pumped out of trucks and blanketed across residential streets in thick clouds, the public perceived pesticides as brute force chemical weapons. But that perception misses the actual mechanism of action. Neurotoxins are not acid. DDT does not melt cells. It does not cause tissue destruction or cellular lysis. It acts as a highly specific kinetic disruptor. To understand how this poison actually works, we have to stop looking at the macroscopic organism and start measuring the nanosecond timing of its internal proteins. Mastering modern neurotoxicology requires shifting our perspective from the biology of the whole insect down to the precise microphysics of a single nerve cell. If we pierce through the exoskeleton and travel deep into the insect's internal anatomy, we reach that specific operational battleground, the axonal membrane. This boundary layer of the nervous system is where the chemical exchange of information happens. Embedded within this lipid bilayer is our central protein of interest, the voltage-gated sodium channel. This transmembrane protein acts as a gated pathway, strictly controlling the rapid influx of sodium ions required to initiate and propagate electrical action potentials across the neuron. The entire survival of the insect nervous system is precariously balanced on the proper rhythmic functioning of this single protein complex. To see how the poison breaks the system, we first need to map a healthy channel's gating cycle. It moves through 3 strict conformational states, starting here in the resting or closed state. When the membrane reaches a specific electrical potential threshold, the protein's structure is forced to change shape. It snaps into the open or activated conformation, allowing a rapid rush of positively charged sodium ions to pour into the intracellular space. This sudden influx of positive charge immediately depolarizes the local cellular membrane, firing the electrical signal down the line. But the most critical feature of this open state is its brevity. Almost instantly, the channel transitions to its third state, inactivated. A structural plug swings shut, physically blocking the pore and halting ion flow, even if the membrane remains depolarized. This spontaneous inactivation during a firing event is distinct from deactivation, which is the channel resetting to its resting state only after the cell repolarizes. This rapid, spontaneous inactivation serves as the essential biological brake. Without it, there is nothing to prevent a runaway cascade of electrical firing. Now, enter the xenobiotic agent, the chemical dichlorodiphenyl trichloroethane. Because DDT is highly lipophilic, it partitions into the fat-rich axonal membrane. Once inside this lipid environment, it encounters the transmembrane sodium channels embedded there. Here is its primary mechanism of action. The DDT molecule binds directly to the sodium channel exactly when it opens. It acts as a microscopic structural wedge, physically locking the protein and artificially stabilizing that open conformation.
This creates a direct competitive inhibition of the channel's normal kinematics. Because the wedge is lodged inside, the channel can neither deactivate nor spontaneously inactivate. The plug cannot swing shut. The biological consequence is an unending flood of sodium ions pouring through the locked pore. This persistent influx forces the neuron into a permanent state of depolarization. What should be a controlled millisecond action potential devolves into lethal, uncontrolled hyperexcitability. We can observe the resulting tremors in the insect, but neurotoxicologists quantify this lethality directly using strict electrophysiological data. They do this using whole-cell patch clamp recording, a technique that allows researchers to directly measure the ion currents flowing across a single-cell membrane. This chart shows the voltage clamp protocol. An isolated insect neuron is held at a negative resting potential of -90 millivolts to keep all channels closed. Then, the voltage is sharply stepped up to +60 millivolts. This depolarizing step forces sodium channels to open, eliciting a current. In the control trace, we see a healthy channel's response, a sharp downward peak as sodium rushes in, followed by a rapid curve back to baseline as the channel plugs itself shut. Now we test a neuron bathed in DDT. The peak remains, but is followed by an elongated, flat tail. The current fails to return to baseline because channels are jammed open. This failure is quantified as a severe increase in the decay time constant, tau.
A pesticide's lethality is ultimately defined by this highly specific mathematical failure of the sodium current to decay. Because DDT is so highly lipophilic, standard radioligand binding assays struggle to pinpoint exactly where the toxin attaches to the protein. How do researchers map an atomic target site they can't easily isolate? They reverse engineer it using a technique called site-directed mutagenesis. First, researchers use PCR and mutagenic primers to intentionally introduce a highly specific point mutation into the insect's sodium channel gene. Next, they apply a restriction enzyme called DPNI to selectively degrade the original wild-type DNA, leaving only the newly synthesized mutant plasmid behind. This modified DNA is then transcribed and injected into a host cell, often a frog oocyte, tricking the cell into expressing the mutant sodium channel. These mutant cells are placed back into the patch clamp reg and exposed to DDT. When a mutation results in a patch clamp trace that shows a normal, rapid decay constant despite the presence of DDT, researchers have pinpointed the exact atomic target site. This laboratory workflow mirrors reality. That targeted mutation process is exactly how pest populations naturally evolve target site resistance to modern pyrethroid insecticides in the field. Mapping these structural vulnerabilities is a prerequisite for rational drug design. Developing the next generation of highly selective insecticidal agents depends entirely on understanding how to artificially manipulate these precise protein conformations. Securing the global agricultural supply chain means looking past the surface of the insect and decoding the strict mathematical gating of its ion channels.